Okay, now we all know what is the building unit of all organisms. It is the cell. But have you actually ever wondered what is the history of cell discovery? Well, the discovery of the cell is related to the invention of microscope. This means that before the invention of the microscope, scientists weren't able to discover the cell. Okay, we have three principles that we call the cell theory. These principles summarize the effort of all scientists. The first one is that all scientists agreed that all organisms are made of one or more cells. Some living organisms are made of one cell, which are unicellular organisms, and some are made of more than one cells, which are multicellular organisms. Number two is that the cell is the basic unit of structure and organization of organisms. That means that the cell could carry out all biological functions without having to depend on other cells. The final one is that all cells come from pre-existing cells. This happens during cell division. Okay, we mentioned before the relation between the invention of the microscope and cell discovery. So if scientists didn't invent microscopes, we wouldn't have known about the existence of the cell. So let's discuss the types of microscopes. There are many types of microscopes, but we are going to summarize this into two main categories: the light microscope and the electron microscope. The light microscope works by depending on light to magnify different objects. Advantages of the light microscope is that it's small, it's easy to use, it's able to examine living organisms like fungi, bacteria, algae while they're still alive, and samples could be pigmented, which means that they could be colored. Disadvantages of the light microscope is that it has limited magnification power. This means that it could only magnify the sample up to 1,000 times with limited resolution. The electron microscope, however, works by using magnets which aim the electron beam to pass within the cell. Advantages of the electron microscope is that it works by electrons which are very small, so it's able to penetrate or go through the cell easily, giving more details, so it has more magnification power and more resolution. Disadvantages of electron microscope is that it can examine dead samples only and it only gives black and white images. There are four types of electron microscopes.
I want to hear all 4 points.
The first type is the scanning electron microscope, which gives a 3D image for the surface or the cell membrane. The second type is the transmission electron microscope, which gives a 2D image for the internal structure of the cell. The third type is the scanning tunneling microscope. This one works by the releasing of electrons from the probe to the space between the probe and the sample. The electrons will be able to make an image for the atomic level, so we could use this kind of microscope to form an image for the DNA. The final type is the atomic force microscope. This one calculates the atomic force between the molecules in the cell and electrons from the probe. All these efforts in making microscopes led us later to classify the cells into two major types, the prokaryotic cells and the eukaryotic cells. But before we know the types, we must discuss something important. We all know that any cell must contain these three structures: the DNA, the cell membrane, ribosomes, and finally the cytoplasm, but this isn't really a structure, it's a fluid that fills the space inside the cell. We are mainly going to compare the prokaryotic cell and the eukaryotic cell by its structure. Let's start with the prokaryotic cells. The prokaryotic cells are very small in size and they have very simple structure. They contain no nucleus, they just have a circular DNA or chromosome which swims in the cytoplasm. An example for prokaryotic cells is bacteria. Then we have the eukaryotic cells, which contain nucleus, organelles, they are bigger in size, and examples for eukaryotic cells, all animal cells and plant cells. Now that we have mentioned the types of cells, we're going to move on to the final point that we're going to discuss in this video, the endosymbiosis theory. That's such a pretty long word, but what is the endosymbiosis theory? What happened is that scientists saw that the eukaryotic cells are evolved from prokaryotic cells by engulfing the small eukaryotic cells inside the big prokaryotic cells. And once it goes inside, it starts working as a nucleus or other organelles.